Hey everyone, this is Immune System video number 9. And just looking at what remains, I think I'm going to have to do video number 10. But number 10 will be pretty short. So this is the material that remains in this chapter. So 4 main topics. One is a kind of an overview of adaptive immunity. So we discussed all the steps involved in activating lymphocytes and their effector response. And so I want to sort of put it into a big picture now, and then tie it into one disease just to illustrate the importance of especially the helper T cells in the adaptive immunity and in immunity in general, actually. Okay, and then I want to switch to another topic dealing with humoral immunity specifically. So we talked all about how B cells are activated, and they differentiate to plasma cells which produce antibodies, but we didn't really talk much about how antibodies work or the structure of them. So that's another topic. And then I'd like to say a few words about immune memory. What is exactly, what does it mean to say that the adaptive immunity has memory and memory cells? Exactly what is that all about? And then finally, I want to distinguish between 2 types of immunity, active versus passive immunity. So I think for this video, I'll tackle the first 2 topics, and for the last one, the last 2 on the list. All right, so this slide is the overview of adaptive immunity, and it's the big picture of everything we've been talking about. Humoral immunity is through the B cells, and if there's a foreign antigen that binds to a B cell, it will activate, and with the help of a helper T cell, it will proliferate. And that clonal expansion that results is going to produce plasma cells making antibodies and memory cells. The antibodies are going to do the job in the humoral immunity. They're going to enter the circulation, both blood and lymph. And if they bind to some foreign antigen, they're tagging that antigen for destruction. Okay, the cell-mediated immunity is through the helper T cells and the cytotoxic T cells. Helper T cells get activated by an antigen-presenting cell. And when they're activated, they produce cytokines. And the cytokines are going to help the activation of B cells and proliferation of B cells. And they're going to help activation of cytotoxic T cells. And they're going to help also immune system, innate immune system cells, such as macrophages and natural killer cells. So the helper T cells are extremely important because they're supplementing, they are pumping up, I guess you could say, strengthening all branches of the immune system, humoral, cell-mediated, and innate. And we'll see what happens when the numbers of these helper T cells decrease. Cytotoxic T cells are going to be activated by an abnormal body cell, such as a virus-infected cell or a tumor cell. And when they're activated, they secrete perforins and granzymes that are going to kill the infected cell and anything that's like it, like any other cell that's infected by the same pathogen. Okay? So 2 of the topics have to deal with things on this slide. One is the central importance of the helper T cells doing all of these different stimuli for the immune system in general. And the other is antibodies. What are they doing? So let's start with the helper T cells first. All right, so the central importance of helper T cells can be illustrated, unfortunately, by what happens when people got infected by the HIV virus when it first appeared in the 1980s. When it first appeared, there was no effective treatment for it, and people unfortunately suffered hugely, greatly from it. There were a lot of deaths. And what happens during HIV infection? By the way, HIV stands for human immunodeficiency virus. So it causes, upon infection, deficiency in the immune system, and your immune system just doesn't work as well. So what some examples of that not working so well are people with an active HIV infection become susceptible to infections by other diseases. So particular types of pneumonia, for example, were characteristic infections. Their immune system wasn't working so well, and they were more susceptible to infection in general. Not only that, they started to appear, certain types of cancers start to crop up in HIV infected individuals. So those are both hallmarks of an immune system that's not working very well. Appearance of cancers and infections. So that's the basis for the name, immunodeficiency virus. Now, how does HIV cause this immunodeficiency? The basis for that is that HIV targets CD4 positive cells. So remember, helper T cells have CD4 as the co-receptor for the T cell receptor. And so CD4 on the surface of the helper T cells is a binding site for the HIV virus. In an HIV infection, the HIV virus attaches to CD4, as shown in this slide. Here's the virus particle, and here's attaching to the CD4 co-receptor. It's actually mislabeled here on the side of the slide. CD4 co-receptor, T cell receptor here. By attaching to it, the virus is then able to enter into the cell and start an active infection in which it will reproduce itself inside the cell. Okay, when it does that, it starts producing new virus particles, and it is eventually going to lyse the helper T cell, and it's going to kill the cell. And initially what happened in the 1980s is people would have an ongoing HIV infection, no good treatment for it at the time, and there would be a steady drop in the number of helper T cells. So normally helper T cells in the blood plasma are somewhere around 1,000 cells per mil, but it can drop to very low levels. Below 200 is when it's given the designation of AIDS, acquired immune deficiency syndrome. And that's where those other infections and cancers start to appear. So without the helper T cells, you don't have this function. You don't have all of these stimulations of other parts of the immune system going on, right? So that gets weaker and weaker. Your immune system is more and less effective through a developing HIV infection. Now, in the 1990s, an effective treatment for HIV came on the scene. And this is a, where is it? It's certain medications. There are 3 of them that are involved in interfering with reproduction of the HIV virus. And people that are HIV positive can now live a relatively normal life taking that medication. The reason why I wanted to talk about HIV infection is just because it illustrates how important the function of the CD4 helper T cells is. All right, let's switch to the other topic, which is antibodies and the humoral response. So I want to talk about antibody structure first, and then I'll talk about what antibodies do when the humoral response is triggered. So antibody structure, antibodies, also called immunoglobulins, are proteins that are produced in response to a specific antigen. So the antibodies have binding sites for that specific antigen. When an antibody is produced, it's able to bind to the specific foreign antigen, and it tags it for destruction by other parts of the immune system. And we'll talk about that in a bit. The antibody molecule is a Y-shaped molecule and it's a soluble protein, meaning it's soluble in blood, plasma, or lymph. There are 4 polypeptide chains in the molecule. 2 of them are identical heavy chains, they're longer amino acid chains, and 2 identical light chains, shorter chains. They're assembled into this Y-shaped structure which has a flexible hinge region. And all of the different chains, 4 of them, are held together by covalent bonds between amino acids. These are called disulfide bonds. There are 2 important areas that are functionally significant, and these are regions that have constant amino acid sequence between different antibody molecules produced by different plasma cells, and regions that vary in amino acid sequence between antibodies produced by different cells. So the variable regions are located at the tips of the Ys, and these contain the antigen binding sites. And these are variable, and that's what accounts for the different ability of different antibodies to bind to a whole host of different possible foreign antigens. The constant regions are sort of the base of the Y, and they're involved in other functions of antibodies, which we'll talk about later. So here is the structure, Y shape, as I mentioned. There are 2 identical heavy chains. Here's one, here's the other. Whoops, where'd it go? Here it is. There's the other, and 2 light chains. These are identical also. Halfway, the light chain is half and half. Constant sequence region, this is constant. This is going to be the same amongst all light chains in antibody molecules. Same for this light chain. The other half is variable sequence. Okay. For the heavy chain, it's about 3/4 constant sequence. Up through here, the lighter shade, that's the same in all antibody molecules. But the other quarter is variable sequence region. The antigen binding site is the tip of the Y. There are 2 of them, and they're made up of the variable region of the heavy chain and the variable region of the light chain. This is going to produce some kind of structure which is going to be complementary in structure to some foreign antigen. For a given antibody molecule, the antigen binding sites are the same. So these are going to be the same for one antibody molecule. All right, here are the covalent bonds holding the chains together. There are bonds that hold heavy to light and heavy to heavy. So let's see, did we talk about everything? The 4 chains, here's the hinge region. So this is about halfway through the heavy chain, flexible hinge region. This is the stem region here. The stem region is the base, and also called the FC region. So as far as the 2 functional regions, the antigen binding site in the variable regions, that's functionally important. And the stem or FC region, the base of the molecule, is functionally important as well. So what do those do? All right, so the action of antibodies is either through binding to the foreign antigen through the variable region binding sites, or through the base of the stem region that's going to interact with other immune substances, either cells or proteins. Okay? When an antibody binds to a foreign antigen, it's going to immobilize that antigen. And it's going to tag it for elimination by other immune system elements. Antibodies are very good for binding or tagging these particular types of foreign antigens. Viruses, bacteria, soluble toxins. Sometimes bacterial infections will produce toxins that they secrete, and yeast spores. All right, so let's talk about what antibodies do through binding to foreign antigens at their antigen binding sites, the variable regions at the tips. So one thing they can do is neutralize a foreign antigen. Simply binding to it can neutralize its pathogenic function. And a really good example of that is if an antigen binds to a virus particle, like HIV that we just talked about, if an antibody binds to that, it's going to interfere with that virus binding to a cell, binding to the CD4 coreceptor of a helper T cell, for example. It's just going to get in the way of that happening. So it's going to neutralize that virus particle just by binding to it and getting in the way. Another thing a virus can do is cause agglutination of a foreign antigen, and that's because there are two binding sites on the antibody. So it can bind to two different antigens, and then it can form this big chain of antibody, antigen, antibody, antigen, and that forms an agglutination, basically a clumping of antigens, and that is going to be a target for macrophages to internalize and break down. This is a very effective way for getting rid of bacteria because bacteria are relatively large antigens and they can have multiple antigenic sites on their surface such that more than one antibody can bind to them. Third thing that can happen when an antibody binds to a foreign antigen is if it's a soluble antigen like a toxin, it can cause it to come out of solution. So what was a soluble foreign antigen that can cause problems, when it comes out of solutions, it's basically no longer active. And it's also going to be target for phagocytic cells to do away with. So those are all illustrated on this slide. These are actions of antibodies through directly binding to the antigen, and it's binding through the variable tips of the Y molecules. So this is illustrating how antibodies can bind to a virus and neutralize its activity. How is it going to gain entry into a cell when it's surrounded by all of these antibodies bound to it? Here's an example of agglutination for a bacterium with multiple antigens on its surface. It can be bound by more than one antibody, and it can form this chain of antibody-antigen. It's just going to form this huge clump. And that's not going to be effective anymore as an infectious agent. Here's an example of precipitation, and this is what most useful for small, soluble, for example, toxins that can wreak havoc in the body. But if they are precipitated by this also antibody-antigen chain of substances here, it's going to take this soluble toxin out of solution, and that's going to make it ineffective. All right, antibodies can also act by virtue of their stem region. The stem region is going to stimulate various cells or proteins in the immune system, and that's going to help with an immune response. How does that work exactly? So here's a couple of ways that an antibody bound to a foreign antigen can act through its stem region. The stem region can activate complement proteins. If you remember back to the innate immunity, complement proteins are present in the bloodstream. They circulate in inactive form. If they're activated, then they're very effective at eliminating or battling bacterial infections. So complement activation can happen through an antibody binding to an antigen and the complement proteins binding to the stem region of the antibody. Another thing that antibodies can do through their stem region is opsonize, coat a pathogen. And this is similar to what complement proteins can do. Both can opsonize a pathogen. And when either of them or both of them opsonize a pathogen, it makes it a much more likely target for phagocytes to recognize, to bind, and to incorporate and degrade the pathogen. Finally, what the stem region can do is activate natural killer cells. If you remember from my discussion of cytotoxic T cells, I mentioned that cytotoxic T cells and natural killer cells act through the same mechanism to kill abnormal cells in the body. Cytotoxic T cells do this by specific recognition of abnormal cells by binding to them. Natural killer cells recognize abnormal cells through antibodies binding to them, and then the natural killer cells bind to the stem region of the antibody. So let's take a look at those three illustrated on this slide. Here is an antibody bound to a bacterial antigen, and then the stem region is going to be binding to a complement protein and activating the complement. Okay, complement would only bind to the stem if the antibody is bound to a foreign antigen. But that's one thing that can happen, right? So this is going to activate complement, and that's going to stimulate the immune response even more. This is illustrating opsonization by antibodies, right? So bound to a bacterial pathogen here. The stem region is exposed, and that's going to be a target for phagocytes to come along, internalize the pathogen, and break it down. And this is showing how if antibodies are bound to a foreign antigen on the surface of an abnormal cell, then that's going to be a target for natural killer cells to latch on, and they're going to be activated. And remember, when they're activated, what they do is secrete perforins and granzymes, and this is going to cause the abnormal cell to undergo apoptosis and to die. All right, well, so that's a pretty good summary of antibody structure and antibody action through either direct binding to the antigen at the antigen binding site or through the stem region. So I'm going to end this video here, and in the last video, I'll cover the last two topics on the outline that I showed at the beginning of this video. See ya.